Hello and welcome back to lecture 3 of week 4 of this course, Quantitative Methods in Chemistry. This week we have so far learned about how we can apply statistics to test various hypotheses that we need to test, and we get introduced to the concepts of z-test, t-test, and also how outliers can be identified for a given data set. So today we will get introduced to two important concepts related to this, which is the pooling of the data to get a better estimate of the population, and also how we can apply a paired t-test to compare two methods on a same sample set. So let us get started. So let me first talk to you about the concept of pooling data to enhance predictability. So this concept is illustrated by this Venn diagram. So before that, it is again very important to understand that this pooling of the data can be applied only for samples that are collected from the same population, and these samples, which are denoted by different colors here, for example S1 denoted by blue and S2 denoted by red, and so on and so forth, they have been collected from this same population, which is the blue circle outside, and the different sizes of these S1, S2, S3 and S4 denote that they contain different number of data sets or different readings. So how do we pool all of this data to get a better estimate about the population? So this data set can be collected at different time points, it can be collected by different peoples. So this makes it easier for us to collect a larger amount of data by distributing our resources. Now once we have collected this data, we can utilize pooling of the data to get a better estimate about the population by this pooling protocol. So what is this protocol is what we will learn now, and you also need to understand that by pooling this we improve the reliability of the standard deviation that we estimate through the pooling. So let us quickly go through the protocol of pooling such data sets. So if you are dealing with samples which we call as S1, S2, S3, so on and so forth, and we are dealing with say T such samples, and in each sample the number of readings is N1, N2, N3 and so on and so forth. What we can do is, the protocol for pooling this data is that we can estimate, so our S pooled will take into consideration the individual deviances which have been squared. Now these will be divided by the individual readings in each data set or in each sample. For example, N1 for sample 1, N2 for sample 2, N3 for sample 3, so on and so forth. But it is important to remember that we need to have the degrees of freedom in the denominator. So since we were dealing with T number of sets, we will have to insert T here and subtract this T number of sets from the total number of readings in all the data sets. So, the total number of readings in all the data sets will be given by N1 plus N2 plus N3 plus N4 and so on, and T will denote the number of samples that have been pooled. Let us quickly take an example with real numbers to understand this concept. So, suppose we are dealing with sample 1, sample 2 and sample 3, and they have the readings as 5.15, 5.03, 5.04, 5.18 and 5.20. That means it has 5 readings in the sample 1. Sample 2 has only 3 readings, which come out to be 7.18, 7.17, 6.97. Similarly, sample 3 again has 5 readings as 6.04, 6.02, 5.82, 6.06 and 5.88. Now, these 3 samples have been obtained from the same population, so we need to pool this data, and I will now explain to you how this is to be done. So, if I take the average value of these samples for the sample 1, this comes out to be 5.12. For sample 2, the average value of this sample is 7.11. Similarly, for sample 3, this value comes to be 5.96. Now, if I calculate the individual deviations, so for example, for sample 1 we talk about deviation 1, then these deviations can be squared for calculating the S pooled value, and these squared values will come out as 0.00. So, essentially these numbers are nothing but 5.12, which is the x1 bar value, which is subtracted from the individual readings in sample 1, and the result that we obtain has been squared. Similarly, we can go about calculating the deviation 2 squared, and that will come out to be 0054.0040.0187, and we can summate these. So, this summation will come as 0.0254, while here for the second sample deviation squared summation will be 0.0281. Similarly, for the third sample, the deviation 3 squared value can be generated, and let me quickly fill in the numbers here. So these are the numbers of the deviations squared for sample 3, and when we sum all of these, this number comes out to be 0.0459. Now from these values we calculate the S pooled as square root of 0.0254 plus 0.0281 plus 0.0459, this divided by 5 readings for sample 1, 3 readings for sample 2, and again 5 readings for sample 3. However, since we are dealing with 3 samples, we will have to subtract 3 from here, and as you go through these calculations, you will obtain that S pooled value now comes out to be square root of 0.0994 divided by 10, which is ultimately going to be 0.1. So this S pooled value is a better estimate of the population standard deviation. So I hope you would have understood how this pooling of the data is to be done and what is the protocol to be employed to obtain S pooled value or the standard deviation value for this pooled data set, taking into consideration the various readings in individual samples. Now let us move to the second topic, which is of applying paired t-test to samples which are being analyzed by 2 different methods. So for that, let me go back to the presentation. Paired t-test for comparing 2 different methods which are applied on the same data set. Now again, it is important to remember that a paired t-test can be applied only when the same data set is being compared by 2 different methods. And one example of that is, suppose there is a blood test that is done on a patient. So suppose method 1 shown here is the reference method, and method 2 is a new method that is being developed to estimate the same analyte. So what we do here is we take samples such as sample 1 or 2 or up to 6, and we use these samples to estimate the analyte concentration using method 1 or method 2. Now there can be differences between the 2 methods, and also there can be differences between the individual samples. So the question that we want to answer here is whether the 2 methods, which are now for us the reference and the new method, give us the same or similar results, or there are differences between the old method and the new method. So let us take this example which is shown on the slide, where we have these six samples and we have applied method 1 and method 2 on them and generated the analyte concentration whose numbers are given in the table. So what I want you to note here again is that both method 1 and method 2 are being applied on the same sample, and there can be differences between the individual samples. And the question that we would want to answer, for example, is that at 95% confidence, can we say that method 1 gives different results than method 2? So here you might recollect that since it is only about the estimation or prediction of differences between the two methods, a two-tailed test can be applied. And we will also again have a null hypothesis that the two methods do not give different results, or the results that they generate are statistically similar, and any minor variations between method 1 and method 2 is the result of random fluctuations during the measurement. So now let us apply the paired t-test to compare these two methods. So let me again go back to the board. So again here, please remember that we will apply the same protocol, except that the t calculated value here will be d bar divided by SD by root n. Now d bar here is the average difference between the two methods, SD is the standard deviation of the differences between the two methods, and of course root n, the n is the number of readings being compared. So let us quickly apply this to estimate or predict whether the method 1 and method 2 in our example are generating different results or not. So they will be generating different results if our t calculated is greater than the t critical value at 95% confidence level, which is the confidence level at which we have to make the prediction, and 5 degrees of freedom. So if I look back at the t table, which is given here, this number turns out to be 2.571 for 5 degrees of freedom and 95% confidence level or 5% significance level. So going back to the board, our t critical value will be 2.571 for 95% confidence level and 5 degrees of freedom. So let us do the maths. So what we have are sample method 1 and method 2 being applied to the same sample, and for sample 1, method 1 gave a reading of 500 while method 2 gave a reading of 480. Sample 2, these numbers were 292 and 305. Sample 3, it was 343 and 325. Sample 4, it was 445 versus 460. Similarly, sample 5 and sample 6 have these readings as 480 and 500. and 395 and 370. So, what we do is we calculate the difference between the two methods, and the difference will come out to be plus 20 in the first case. So, method 1 reports more than method 2. In the second case, method 2 is reporting more. So, this difference becomes negative 13. Similarly, for sample 3, this is positive 18. Sample 4, it is again negative 15. It is also negative 20 because method 2 is reporting more. And finally, for the sixth sample, the value is plus 25. So, if we sum up all these differences, then the total sum of the differences is plus 15, taking into consideration all the signs that are present. We also do the difference squared, and that number comes out as so. How does this number of 306.25 comes? So, what we have done is we have calculated the average difference, which is nothing but 15 by 6, which is 2.5, and for the first reading we have subtracted 2.5 from 20 and squared that number. So, 17.5 squared will be 306.25. When we do this exercise down the table, the numbers turn out to be 306.25, 506.25, and again 506.25, and you can summate all of this to 2105.5. So, from these numbers we will be able to calculate the standard deviation of the differences. SD value will be square root of and when we plug in these values, this would be square root of 2105.5 minus 15 square, that is 225, divided by 6 and all divided by 5, and square root of deviations squared by n minus 1, which is nothing but 2105.5 divided by 5 square root, and this number will ultimately come out to be 20.52. So, our standard deviation of the differences between the two methods is 20.52, and the average difference was 2.5. With these, let us calculate the T calculated value, which is d bar by SD into root n. Now, d bar is 2.5 divided by 20.52 into square root 6. This upon solving will come out to be 0.298. Now, going to the next page, we have the T calculated value as 0.298, while our T critical value was 2.571. And remember that our null hypothesis here was that method 1 gives similar results as method 2. And when we test the null hypothesis here, we observe that the T calculated value is actually less than the T critical value. That means the null hypothesis holds true at 95% confidence level, or method 1 and method 2 are giving us statistically similar outputs or similar results. So, you can see that you can apply this pair T test on methods which are being applied on the same samples. Now, I have also collected a couple of other questions for us to discuss, which will sort of summarize our understanding of this course for this week. So, let us quickly go to these questions. So, let us take this example of a battery company that warranties its batteries for 48 months, so that only 0.5% of its batteries fail within this warranty time period. However, immediately after the warranty time period gets over, that is in another 6 months, there is a significant reduction in the number of working batteries, and 2.5% of the batteries fail to perform. So, with this data, the question that we are dealing with is, what is the average life of the batteries being manufactured by this company? So, let us go back to the board and understand how this problem is to be approached. Now, here, since we are only concerned with batteries that fail during the 48-month or 54-month period, we are concerned only with the one tail of the Gaussian profile. So, we are only concerned with this portion of the Gaussian profile. Any battery that keeps performing beyond 48 or 54 months is considered to be good. So, in other words, when we say that 0.5% of the batteries are failing, we are essentially implying a significance level of 1%. So, just to repeat this point, we are dealing with a one-tailed test here. So, what we get from this data is, if mu is the mean value of how long the battery lasts, then mu minus z sigma in this case is 48 hours, 48 months, when the z value in this case is 2.54, corresponding to a significance level of 1% in the two-tailed test. So, if we plug in these values, we get mu minus 2.54 sigma is equal to 48. Similarly, since 2.5% of the batteries are failing, then the z value in the second case will come down to 1.96, which implies that a total of 5% of the significance level for the two-tailed test. So, where mu minus 2.54 sigma is 48 months, and another where mu minus 1.96 sigma is 54 months. So when we solve these two equations, what we get is that sigma is equal to 10.345 months, and when we plug this into mu minus 2.54 into 10.345 is equal to 48, which was our equation 1, then we obtain the mu value as 74 months approximately. So 74 months is the approximate life of the batteries being manufactured by this company, and with a standard deviation value of 10.345 months. And if you have these numbers, the company can warrant its batteries for 48 months with only 0.5% batteries being failing in this time. Now let me take another question here, which corresponds to how medical tests are performed and how inferences are made through them. So typically when we undergo a medical test, say a blood test or a urine test, there is a normal range which is specified, and any numbers greater than or lower than these normal range are considered to be suspicious. But being suspicious does not always mean that there is a problem with the sample or the patient from whom this sample is taken. So in this question, there is a medical test presumed to be in normal range. If it lies in the 95% confidence interval, that means 95% of the population will have its numbers within this range. And if a sample's registered low out of range value, how likely is this sample to indicate a disease that inflicts 0.1% population? So suppose there is a disease which we know through previous studies that it inflicts only 0.1% population, and it gives also in the patients who have these diseases, there is a particular analyte whose value comes out to be low. So we conduct a blood test on a patient, and we indeed find that the blood sample gives out a low out of range value. Do we say that this sample is coming from the patient who has a disease or not? Or how likely is the patient to have the disease just because this number turned out to be low? So let us do our calculations quickly. Let us go back to the board, and here what we know is that 95% population would give the in range reading, and 5% of the population will give out of range reading. Now this out of range can be either on the left side or on the right side. So the numbers can be significantly low, or the numbers can be significantly higher than the prescribed range of values that they should have. So in this case, the question says that the patient had a low value. That means we are talking only about the left tail of this Gaussian profile. So in other words, there is only 2.5% population that will give out a lower reading than the normal values. Now this 2.5% population is that which will give out a low reading, and only 0.1% population will actually have the disease. So the probability of a person giving a low reading and also having the disease would be 0.1 by 2.5 or around 4%. So only in 4% of the patients who give low reading of this analyte can there be actual disease. So I hope you understood how we apply statistics to understand how blood tests are performed and how predictions are made through these tests. So this brings us to the end of our 4th week of classes for this course, and in the next week we will be discussing about how analysis of variance is performed on a large data set. Thank you.